Dear students, today the laws of Mendel is going to be the topic for our discussion. I have already told you Johann Gregor Mendel, the father of genetics, after a very hard work, after having performed a number of experiments in Pisum sativum plant, taking 7 characters for his analysis, came out with a number of laws. He took 7 characters for his study, and at a time he was concentrating only on one character. After his monohybrid experiments were over, then he conducted experiments based on taking 2 characters into consideration called dihybrid cross. So, by doing the monohybrid and dihybrid cross, he came out with a number of laws. These laws are popularly called as laws of Mendel in genetics. So, based on his experiments of monohybrid and dihybrid crosses, Mendel proposed 3 important laws which are now called Mendel's laws of heredity. Now, even though I have put 3 here, the number differs. Actually, here I have listed 4. Even in the textbooks, it differs from 3, 4, sometimes even 5. It all depends upon how they state the law. For example, law of unit characters and law of dominance and recessive may go together. In that case, you will be having only 3 laws. Law of segregation will be there, and then law of independent assortment will be there. Sometimes they quote law of segregation and law of purity of gametes as a separate laws. Whatever it is, the laws of Mendel are always laws. The number differs depending upon the way in which it is being stated. Okay, right. So we will take up the discussion on one after another. The first one is the law of unit characters. Now, what do you mean by the law of unit characters? This law states that each and every character is controlled by a pair of factors. Now we call them as alleles, a pair of alleles. So, each and every character is controlled by a pair of factor is the first law or the law of unit character of Mendel. Now, how do we come to this conclusion, or how Mendel came to this conclusion? When he was doing experiment on the Pisum sativum plant, he came across the results with which he was able to clearly and categorically tell that each and every character is controlled by a pair of factors or a pair of alleles. Now, what do we mean by this law? In a particular homologue, if you take in a particular locus, let us imagine the gene for the height of the plant is there, representing the tall and the dwarf characters or traits. So, to represent the tall character, you have got a pair of factors. These 2 factors could be in a homozygous form or it could be in a heterozygous form, and the dwarfness is represented by 2 factors. So, each and every character is controlled by a pair of factors. It cannot have more than that, or it cannot have less than that also. But there are certain exceptions. That's what we are going to discuss that in a detailed way. First, we will discuss, Mendel has told that it is each and every character is controlled by a pair of factors. Now, just for the sake of argument, let us imagine that I am arguing that a character is not controlled by a pair of factors and it is controlled by only one factor. Okay, let us imagine in that way. So T represents the tallness. T represents the tallness, and this represents the dwarfness. Okay, fine. Now what Mendel did is, Mendel took a tall variety and then crossed it with a dwarf variety. So tall is represented by like this and dwarf is represented like this. Now as a result of this cross, he got all tall in the F1 generation. So this tall plant should also be like this only. If we are going to imagine that each and every character is controlled by a pair of factors, only one factor. Now if this is self-fertilized, then it will be like this, and there is no possibility for this combination to give rise to a plant of this nature, which we have imagined for the dwarf variety. Okay, when you make a cross, you get tall and the dwarf in the ratio of 3:1, and if you imagine that each and every character is controlled by only one factor and not two factors, as I have described here, you cannot account for what is happening in the Mendel's experiment, because in the F2 generation for monohybrid, the phenotypic ratio is 3:1, that is appearance of the dwarf plant once again in the F2 generation. So if this plant has to appear in the F2 generation, which is getting masked in the F1 generation, so this factor should have been masked in the F1 generation. So if it is getting masked, means each and every character must have only two factors. So it is to that conclusion we have to come. So the first law of Mendel states that each and every character is controlled by a pair of factors. Okay, fine. Now, do you have any exception for this, or this is a universal law? We will come to that point. Do you have any case where a character could be represented by less than two factors? Do you have any condition like that? Yes, it is there, because in haploid organisms, namely algae, fungi, and in bryophytes, or the plants having the gametophytic generation, there will be only one set of chromosome. N number will be the number of chromosome. So each and every character cannot be represented by a pair of factor, and it can have only one allele or one factor. So in haploid organisms like algae, fungi, bryophytes, in the gametes, wherever there is a monoploid is there for a particular character, it could be represented by only one factor and not two factors. So there is a slight deviation, or there are some certain exceptions for this law, namely the law of unit characters. But by and large, generally it is accepted that each and every character is controlled by a pair of factors. Okay. Now coming to the second point. Do you have any character which is controlled by more than two factors? Second point. Do you have any example to prove that each and every character is controlled by more factors? There also the answer is yes. There are many characters which are controlled by many factors. Now this is what is called as a multiple allelism. Multiple allelism. What do we mean by the word multiple allelism? See, each and every character is controlled by many alleles, many factors. Now, till the period of Mendel, there was no incidence where he could observe that a character could be controlled by more than two factors. So he told that always the characters are controlled by a pair of factors. But after the period of Mendel, when people like Shermer, Corins, and all these people were working on the plant and animals, they came across a number of situations where a character could be controlled by not just two alleles, many alleles could be there. It is to that conclusion they came. So there opened a new chapter in the books of genetics, what is called as a multiple allelism. Today we have got any number of examples for multiple allelism. The beautiful examples we are getting for multiple allelism, you have got it in plants, animals, in human being, everywhere you get an example. In plant, there is an example where the self-sterility nicotiana, a character called self-sterility in nicotiana. Now, this character is controlled by at least 7 alleles, they say, and then they designate it as S1, S2, S3, like that, S7. S stands for the word sterility. So a character called self-sterility nicotiana is controlled by minimum 7 alleles, they say. So there is a beautiful example for multiple factor inheritance or multiple allelism. In animals we have got a number of examples. The coat color of a rabbit, it is controlled by 4 alleles, and these 4 colors are called as agouti, chinchilla, Himalayan, and finally albino. So these 4 characters or traits representing the coat color of rabbit is controlled by 4 alleles or 4 factors. So once again you have got an example to show that many factors could be there for deciding or determining the character. Now in the animals, the maximum number of the allele that we come across is in the case of the eye color of Drosophila. See Drosophila fly. Here in this Drosophila fly, the eye color of the eye is decided by at least 14 alleles, they are telling. So the most dominant color is red, and it is going on changing to crimson red, eosin red, brown, and then dark brown, light brown. It goes like that, and then finally it comes to white, the 14th color. So starting from red ending with white, the eye color of Drosophila is decided by at least 14 factors, they are telling. This is a maximum number of the factors that a gene could have, they have identified. A beautiful example of multiple allelism you come across in the human being also. See, the blood group itself, the ABO blood group itself is the best example for multiple allelism. It is containing 3 alleles, namely A allele, B allele and O allele, deciding the 4 groups, namely A group, B group, AB group and O group. Okay. So these are all some of the beautiful examples of our multiple allelism. So to sum up, what I mean to tell is the first law of Mendel, the law of unit characters, is not universal, first point. But whatever examples Mendel came across, it is all about the 2 factors. So he made it as a law that each and every character is controlled by a pair of factors, even though at a later stage people found out that there are many cases where a character could be decided by more than two factors. Okay. Now that completes our discussion about the first law, namely the law of unit characters. The second law is a law of dominance. The law of dominance and recessiveness states when two homozygous individuals with one or more sets of contrasting characters are crossed, the characters that appear in the F1 hybrids are dominant and those who do not appear in the F1 are recessive characters. It could be more, it could be restated in a more elaborate way also, that we will discuss in the slides that are going to further come. So this is the law of segregation, the next law, but we will go for the discussion of the law of dominance. So what the law of dominance states, when you take a pure breeding variety of a tall plant and then cross it with a dwarf variety, you get all tall in the F1 generation. And when these all tall plants, hybrid tall plants are crossed, you get tall plants and dwarf plants in the ratio of 3:1. So what it is stated is, when two pure breeding varieties differing in one essential character is taken and then crossed, the character that is appearing in the F1 generation and 3/4 of the F2 population is called as dominant. You can put it in that way. Okay, I will restate. When you take two pure breeding varieties differing in one essential character, you take, and then when a cross is made, the character that is getting masked in the F1 generation and reappears in 3/4 of the F2 population is called as a dominant. Now, the character that is getting masked in the F1 generation and reappears in the F2 generation is called as recessive. So this is called as a recessive. So dominance and recessive. So once again I am repeating this law. When you take two plants or two animals or two organisms differing in one essential character, the character which is getting masked in the F1 generation and appears in 3/4 of the F2 population, and the character that is masked in the F1 generation and reappears in the 1/4 of the population is called as recessive. Okay, so this is what we mean by the law of dominance. Fine. Now we will go to some more discussion about this law. Is there any exception for this law? For the law of dominance, do you have an exception? Now there are certain exceptions. There are certain exceptions. We understand that there are certain exceptions. As I was discussing earlier, till the time of Mendel, he did not come across any situation where there could be some exception, where there could be a case where the dominant character is not appearing. He never came across such a situation. So he made this as a law called law of dominance. But later on, when people were doing work, particularly when Corinth was doing some work on the flower color of a plant called Mirabilis jalapa, Mirabilis jalapa, Mirabilis jalapa, called as a 4 o'clock plant. or Andimali in Tamil. It belongs to the family. It belongs to the family Nyctaginaceae. Nyctaginaceae. Then, have you taken down this information? Good. When a red color flower is taken and then crossed with a white color variety, you get pink in the F1 generation. Okay. If red is going to be dominant over white, red must have appeared here. If white is a dominant over red, then white must have appeared here. But actually speaking, pink is appearing. That means the red is not a dominant over white, or the white is not a dominant over red. So he preferred to call this a particular case as incomplete dominance. Incomplete dominance. Okay. So when this pink variety is once again inbred, self-fertilized, you get red, pink and white in the ratio of 1:2:1. So this is a beautiful example of incomplete dominance, where there is no complete dominance. Red is not dominant over white and white is not dominant over red. So many cases are there like this. Hundreds of examples today we have met with this. Particularly when it is going along with the lethal allelism, incomplete dominance is working very well. Okay. So there will be three categories always. There is not two categories as in the case of Mendel's case. There will be always three categories. Okay, like this. And the law of dominance, as I was telling you, is not an universal law, and you have got exception for this. And one such case is a law of, the case of Mirabilis Jalapa. But anyhow, the law of incomplete dominance is not giving you a wrong information that these two colors will get mixed, and then you will be getting a pink, a third color called pink. So here actually there is only the mixing of the effect and not the mixing of the factors or mixing of the alleles. Now if these two characters have mixed together to produce a pink color, then there is no possibility for you to get a white color from this pink color, because it has been already mixed. So there is no mixing of the alleles or mixing of the factors, but it is only the mixing of the effect. Okay. So that's what is called the law of purity of the gametes, which we will be discussing in the along with the law of segregation. So the third law is a law of segregation, law of purity of gametes. Now what do you mean by segregation? Segregation means separation. So it gets separated. So this law, though it was a conception originally and propagated by Mendel, now it has been confirmed by cytological studies. Segregation holds good for all cases. That's what they have given. Segregation holds good for all cases, because the previous two laws, namely the law of unit characters and the law of dominance, they have got their own exceptions. But this law of segregation is an universal law, and you don't have any exception for that. We will discuss about that in a detailed way. What do you mean by the law of segregation? As we were discussing, first we will go to this is a monohybrid cross. Once again I am taking the case of a tall plant, a hybrid tall, as it is shown in the F1 generation I have taken. Okay. So this tall plant, while it is inbred, do you further proceed? You write like this. First you draw capital T, small t, and then here once again capital T and then small t. This is a male plant, so these are the pollen grains, and this is a female plant, so these are the eggs. So let us imagine these two are the eggs and these are the pollen grains. This will have a chance of being deposited over this egg, giving rise to capital T. This with this egg giving rise to capital T, small t. This pollen grain with this egg giving rise to capital T, small t, and this with this giving rise to a dwarf plant. So you are getting 3 tall and then 1 dwarf. Now if you are going to accept that this is the condition, then you see these two factors, the capital T, the dominant factor and then the recessive factor, both these factors they segregate and separate, they go to the two different poles. Now if you imagine once again that there is no segregation, you cannot account for what is called as the formation of the characters in the subsequent generation. Now let us put it in this way. So the tall is represented by this and the dwarf by this condition. Now let us imagine that there is no segregation or separation, there is no reduction division. So this will give rise to a pollen grain containing both the factors. This will also give rise to an egg which is containing 2 factors. So egg is containing 2 factors, the pollen grain is containing 2 factors. When this is deposited over this one, then this will contain 4 factors which will be called as a tall plant. So here this tall plant is represented by 2 factors and this tall plant is represented by 4 factors. How is this possible? So logically we are not able to come to the reason for the appearance of a tall character here. Once again in the subsequent generation, if this is going to produce a pollen grain, that pollen grain will have 4 factors like this, and if it is going to find a place over another stigmatic surface, then it may become like this. So the number of factors deciding a particular character will be going on increasing generation after generation. So segregation or the separation should take place definitely at one stage of the life cycle. This is what Mendel told as a law of segregation. Law of segregation is an universal law, and if that is going to be that could be presented in another way also. See, this is a homologue with a pair of chromosomes, and then it is containing a particular character. Let us put it in this way. Now at the time of pollen genesis, or at the time of spermatogenesis or oogenesis, whatever it is, or at the time of reduction division, or the time of fertilization, you may use any term. So or at the time of the meiosis, these two will go to 2 different poles. You see, they get separated. Similarly, the other plant, other chromosome, it will go to one pole and this will go to another pole, like this. So if it has to be represented by a diagram like this, this is one chromosome, this is another chromosome, this will go to this pole and this will go to this pole. Are you able to follow? So separation of a maternal gamete from a paternal gamete is a rule. If they do not separate and then go out, this and this will not be able to combine together to produce a dwarf plant. So they get separated and then reunite. So the separation is a law of the nature. Are you able to follow? Okay. Now the human being is containing 2n number is equal to 46. A man containing 46 chromosome is marrying a woman who is also containing 46 chromosome. Now if the chromosomes are not getting segregated, and then if during the reproduction when they unite together, if no reduction division means the zygote will be containing 92 chromosomes, which is not possible. Now if this baby is going to contain 92 chromosomes, when he or she grows into an adult and then he is reproducing, 92 plus 23 will once again come to different numbers. Okay, 46 to 20, I'm sorry, when there is no reduction division, it will combine with 46. See, this many number of chromosomes will be formed. It will be going on changing from generation to generation. It will be getting doubled. Are you able to? So since reproduction is considered, sexual reproduction is considered to be a union of two individuals, namely a male and a female, there should be a sort of reduction division by which the chromosome number is halved. So when a man with a 46 chromosome is reproducing, his sperm will be containing 23 chromosome, and the woman with a 46 chromosome, the egg will contain 23 chromosome. The union of this one will result in a restoration of the 46 chromosomes once again. This is what we mean by the law of segregation. Law of segregation means a law of separation. The 2n number of chromosomes, it is getting reduced to half of the number. Here also it is getting reduced to half of the number. When it is getting reduced, the chromosomes segregate or separate, and then they go to the two different poles. So this law of segregation is a universal law and there is no exception for that. Okay, fine. So that completes our discussion about the third law, or the law of segregation. I am going to give you a recap of what I have done so far. So far we have discussed three laws: a law of unit characters, a law of segregation, law of unit characters, law of dominance, and then the law of segregation. The law of segregation, sometimes it is also stated as a law of purity of gametes. Okay. Now then we are going to the third or fourth law, the law of independent assortment. Now when we discuss about the law of independent assortment, we should always remember that the law of independent assortment can be proved only in a dihybrid cross and not by a monohybrid cross, because the chromosomes assort in an independent way. That's what the law is telling. We'll go for the discussion of some time. The law of independent assortment. In case of inheritance of two or more pairs of characters simultaneously, the factors or genes of one pair assort out independently of the other pairs. Mendel gave this law based on his dihybrid cross experiment. Here the total number of individuals in F2 will be 16, which occur in the ratio of 9:3:1, where two parental classes and two new combinations will be produced. So this is what is called law of independent assortment. Now let us try to understand this experiment very clearly. Now law of independent assortment, as I was telling you, is proved with the help of dihybrid cross. Dihybrid cross means we take two characters simultaneously. So I am taking an yellow round plant and then cross it with green wrinkled plant. Okay, green wrinkled plant means what? The plant is not green and it is not wrinkled. It's not like that. So the color of the cotyledon is green and the shape of the fruit is, the seed is wrinkled. It is in that way. Okay. So an yellow round variety is taken and then crossed with a green wrinkled variety, and then you get yellow round variety in the F1 generation. You get yellow round variety in the F1 generation. Capital Y, small y, capital R, small r. Okay. Now, when this is one plant, when this is self-fertilized, pollen is taken from this plant and then deposited over the stigmatic surface of the same plant. So this will be producing 4 gametes and that will be also producing the 4 gametes. How to write the 4 gametes? See how I am writing. This is one gamete, that is a capital Y, capital R. While these two chromosomes are going to one pole, the leftover chromosomes, namely small y and small r, will go to another pole. Now, these two chromosomes may also have a chance of going to one pole, namely capital Y and small r. When this is taking place, these two will go to one pole called small y and capital R. Now, only when you accept this, only when 4 gametes are produced in this way, 4 gametes on the female side, 16 zygotes could be produced, which will fall in the ratio of 9:3:3:1. So, when Mendel got a 9:3:3:1, his brain was working in the, what is known as the backward, deriving it in the, from the bottom. 9:3:3:1, when added, you get 16. Now, this is 16 is a fertilizer product. 16 is the total number of fertilizer product, which is a union of 2. So, what is the square root of 16? It is a 4. That means 4 male gamete and 4 female gamete must have been produced. It is to that conclusion he is coming. If 2 characters have to produce 4 gamete means an independent assortment should be there, should have been there. It is to that conclusion he is coming. So, when capital Y and capital R are going to one pole, small y and small r will go to one pole. Capital Y and small r may also go to one pole with small y and capital R. Now, that means a capital Y and capital R are not bound together. They are not depending on one another. See, a capital Y chromosome may move with the capital R, or it may move with the small r also. That means the movement of one pair of chromosome is totally independent of the movement of the other pair. So that is what is called the law of independent assortment, because it is independently moving. It is independently moving. It's called a law of independent assortment. Now, for the formation of the gamete, that is a general law by the law of independent assortment. It is 2 power n for the gamete. Monohybrid, it is like 2, and dihybrid, it is a 4. That is capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are the 4 gametes. Okay. And for a trihybrid, it is 8. So, how we calculate this? It is by means of what is called as a 2 to the power of n. Now, in a human being where the n number is equal to 23 pairs of chromosomes are there, how many type of sperms will be produced in each human being, you can very easily understand. It is a 2 to the power of 23. It is a quite an unimaginable number. So even in a man, even in a well-grown man ready for reproduction, when the spermatozoids are produced, billions and billions and billions of spermatozoids are produced in the body, but type wise, genetically, each one is different from the another. The spermatozoid once produced will have the same genetic information. It would have never occurred earlier, and it is not going to happen in the future also. Each and every spermatozoid is unique in its own way. That's what we are coming to that conclusion. So 2 to the power of 23 type of spermatozoids will be produced, and 2 to the power of 23 type of zygotes, egg will be produced. So this is accounting for what is called as variability. See, we are always studying about the biodiversity, variability. In botany and zoology, in biology, the variation is a law of the nature. How this variation is formed, how new organisms are produced in the nature, it is because of the independent assortment. Every time when the chromosomes are assorted or going to the two different poles, it will be having different. One time this and this will go to one pole, and it may go in any way. Okay, as a result of that, 2 to the power of 23 times 23 type of spermatozoids are produced, and a combination with that of 2 to the power of 23x, it will result in 2 to the power of 46 combinations are possible, which is accounting for what is called as a variability in nature. Not two human beings are very same or similar except the identical twins. How it is happening? It is because of this independent assortment. Okay. Now you may be having a doubt in your mind. So this part of that is also very important. I am just drawing the diagram of F1, capital Y, small y, capital R, small r. So this is how it is. The chromosomes are arranged in the F1 generation. Now my argument is, I told that the capital Y and the capital R may go to one pole, with small y and small r going to another pole. Capital Y and capital R going to one pole, and small y and small r to the other pole. Fine. I told that these will also have the chance of going to the capital Y and small r. Now the problem is, you may not be able to very clearly understand that when the spindle fibers are coming from the pole, and then when it is going to drag the chromosomes to the two different poles in this way, obvious that a spindle fiber will go and attach it to the capital Y and then go all the way to the small r to drag it there. See, is it possible to for the capital Y chromosome and the small r chromosome to go to the same pole? You have to imagine the things in a three-dimensional way. See, it is not the way in which it has been drawn here. Actually speaking, they have been arranged in cis and trans method, which you will be understanding, or your teacher will teach you somewhere in the next stage. So the chromosomes are arranged in this method, that is a capital Y, small y, capital R, small r. Now capital Y, small y, capital R, small r. See, now you see. Actually, it is not that the spindle fibers are piercing through something else and then it is reaching it. It's not at all like that. See, when the independent assortment is taking place in this cell, a cell of this nature, capital Y and capital R will be one gamete, small y and small r will be other gamete. When it is taking place in this type, capital Y and small r will be one type, small y and capital R will be the other type. Okay. So, the change is taking place in the arrangement of the chromosome itself. But Mendel was understanding it in a different way. He told that it is an independent assortment. The chromosomes have got the ability of the movement which is independent of one another. So, that is what we mean by the law of independent assortment. Fine. So, that completes our discussion on what is called as the laws of Mendel. The laws of Mendel are very beautiful, very systematically analyzed and understood. As I was telling you in the very beginning, the number of law is not very important. It all depends upon how it is being stated. The law of unit characters, one law of dominance, second law, the law of purity of the gametes, third law, the law of segregation, fourth law, the law of independent assortment, fifth law. Like that, they will be giving in different forms and different versions. But the crux or the essence of the point is this: the law of unit characters, which states that each and every character is controlled by a pair of factors, and these factors could be in a homozygous form or it could be in a heterozygous form. If it is in a homozygous form, it could be a homozygous dominant or homozygous recessive. If it is in the heterozygous form, one will be dominant, another one will be recessive. When you take two organisms differing in one essential characters and then make a cross, the dominant will appear in the F1 generation, and in the 3/4 of the F2 population, the recessive character is getting masked in the F1 generation, and then it is reappearing in the 1/4 of the F2 population. And the chromosomes are getting segregated and separated at the time of gametogenesis or sporogenesis or oogenesis or pollengenesis, or at the time of meiosis. It could be stated in any form or any way. Then finally, the law of independent assortment, which states that the movement of a pair of chromosomes is totally independent of the movement of the other pairs of chromosomes, and it could be taking place in a variety of ways. This is what the law of independent assortment states. So, it is because of these laws of Mendel we are able to understand the genetics in a very detailed way, and then we are able to come to the problems which is existing in the mind of the man quite interestingly, because the one very pertinent question which was in the mind of the man is how so much of variations are produced in the nature. So, it is because of the independent assortment. When the chromosomes are moving in a different fashion, genetically different type of spermatozoids and different types of eggs and different type of combinations are produced in the nature. Okay. Now, that completes an elaborate discussion about what is called the laws of Mendel, and with this the discussion about the classical genetics is over. Okay. Thank you very much for your patience to hearing. Thank you.